Hello, everyone. Thank you for attending today's webinar, Precision Medicine in Clinical Development, A Current State of Affairs, presented by Medidata. I'm Joe Keenan, contributor, editor for Fierce Markets, and I'll be moderating this webinar. Our speakers today are Jeff Weiser, Genomics Portfolio Director, Metadata Solutions, and Ruthie Davey, PhD, Director of Data Science, Metadata Solutions. You can read their full bios on the right side of your window. I'd like to just go over a few technical notes before we begin. If you'd like to download the slide deck, please click the Resources List button at the bottom of your screen. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your volume is up. You can find additional answers to some common technical issues located in the Help button at the bottom of your screen. The webinar is being recorded and will be available on demand within 24 hours after the event. We will follow the presentations with a Q&A session. Please feel free to submit your questions using the Q&A button on the left side of your screen during the presentation. I think that pretty much covers it. We're about ready to begin. Jeff, please go ahead.
Thank you very much. Thank you for the opportunity to be here. I'm actually fairly recently hired to Medidata. I joined about 4 months ago. For the last 10 years, I was privileged to lead an NIAID-funded effort in collecting, QCing, curating, sharing, and analyzing publicly available immunology research and clinical data, which was located and still is located at a website called Import. This was a collaboration with a bunch of academics, particularly the PI at Tool Butte and a bunch of great immunology researchers at Stanford. And it really was a great project to help understand the difficulties in dealing with this combination of clinical trial data sets and these newer assays like next-gen sequencing, but even in addition to that, high-throughput cellular and proteomic assays as well. Now, I have to say I was pretty intimidated facing a presentation on a topic like precision medicine. Given how overused the term has become, admittedly, and given how many luminaries in medicine and science have weighed in on the topic previously. On top of that, more or less every day, there seems to be some new discovery or announcement or claim relevant to precision medicine and genomics. I'm sure almost everyone heard about the work recently discussed in China where they've cured a blood disorder, beta thalassemia, in an embryo by correcting a single base pair error. And I guess that's what it really comes down to, is that's sort of the ultimate precision medicine. So to try and frame this a little bit more, I looked for a good definition, which is always a good place to start. So this is one I found from the Precision Medicine Initiative, an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle for each person. Okay, that sounds good. But in some ways, precision medicine or patient subsetting or stratification, tailoring, whatever you want to call it, has been around for a long time. We're all pretty well aware of blood type testing when you donate blood to the Red Cross, or HLA typing being performed for organ transplantation. So what has changed? And in short, it comes down to cost and capability. I saw a recent Twitter chat about how it's required to include this slide in any PowerPoint about genomics or precision medicine. So I guess I've now successfully done a webinar on this topic. But that being said, it does prove the point pretty clearly that there have been dramatic changes in this field since 2000. The cost per genome to sequence a genome, that is, DNA or RNA for that matter, is reduced on a rate exponentially faster than Moore's law. And we all know how quickly our phones can act and behave now compared to how they could in 2000. And it's becoming really cost viable to run large-scale studies or even population-level studies on human genomes and to sequence the entire exome or genome. So this really does lead to, whoops, my apologies, I got a little too trigger happy with the button there. So again, I wanted to try and help to center this discussion a little bit more, and I found this additional summary from the Precision Medicine with goals from the last administration, website, White House Archives, whatever your political leanings are, And in these first two bullets, I think it looks a lot more like something that Medidata as a company, Ruthie, and I can speak about. First, we're talking about issues around accelerating the testing of treatments tailored to individuals or groups of patients, whether using genomics or other measures, proteomics, cellular, or even physiological data, or mobile data for that matter, in trials. And second, about supporting the expanded use of genomics in oncology trials, although I would add, of course, there are plenty of other indications that also can benefit from this sort of thing. So I'll focus today primarily on how patient stratification, primarily but not exclusively with genomics, is being integrated into sort of the scientific side of the trial workflow. And Ruthie's going to focus her portion on how the FDA is viewing targeted therapies and how incorporating these therapies into clinical trials is changing trial design. So just to give a little bit of background on Medidata, if you're not aware already, as a company, Medidata has quite a bit of background in developing software and in supporting clinical trials. You can read the slide as well as I can, but the numbers that always pop out to me are greater than 900 customers, 13,000 studies already in the platform, 3.7 million subjects, 18 of the top 25 pharmaceutical companies, so very diverse. And it's always exciting to see 13 of the top 15 selling drugs in some way were processed using the Medidata technology. I mean, really the reason I joined Medidata 4 months back is this incredible footprint in the industry. And I've seen a real interest in the company in enhancing Medidata software to better support newer trial designs, nouveau assays like next generation sequencing, et cetera. And that just makes it a really exciting place to be. So a quick poll question for you all, just to get a sense of the room a little bit for the webinar. Have you personally utilized genomic or neomic assays, for that matter, to subset patients, stratify patients, whatever term you want to use, in your organization's clinical trials or in organizations that you advise, consult, or support? So I'm supposed to give you 30 seconds to answer a yes/no question, so just pretend the Jeopardy music is going now. I won't sing it, I'll spare you. I'm probably about 20 seconds in, give or take. Since it's yes, no, I'm probably going to jump. So yes, and it looks like for the most part, you guys are fairly experienced in this domain. So you probably should come up and do the talk for me. And I would look forward to, at the end in the question and answer section, perhaps talking a little bit more about what you guys have seen as well. So I won't waste too much time given you already have this kind of experience, but for those of you who don't remember your bio, genomes obviously play a big role in our health. The inherited genome side of it, the DNA side, those variants can increase your risk for disease, and they can also impact your response to treatment, or for that matter, the impact of the environment on you, which could then downstream affect treatment or disease progression. On the cancer side as well, these somatic mutations can drive the cancer initiation and progression, and they can obviously impact response to treatment as well. For example, the triple negative breast cancer is when a cancer growth is not involved in any way estrogen receptors, progesterone, or human epidermal growth factor receptors. And these are known not to respond to the common treatments. It's a simple example, but it's one that comes up quite often and is in a way a precision medicine scenario, although a lower throughput one. But for the most part, when we talk about genome level sequencing, we're talking about research assays for discovery. And isn't the point of a clinical trial to validate that a drug is safe and effective? Interesting question, and it's something we've been pondering a lot internally as well. Looking at this chart that navigates through a typical trial workflow, since all of you work in, or many of you work in trials already, you already obviously live day-to-day study design and feasibility, study planning and startup, conduct, closeout, etc. And obviously submission to the FDA and all the other work that goes in. So what we're seeing with regard to how this sort of patient subsetting in genomics is going on and affecting clinical trials is generally, clearly in designing the protocol, during the conduct of the trial, particularly in the execution and analysis, clearly, at conclusion, to queue up or influence later trials, submission to the FDA or other regulatory bodies. And now I think there's going to be an increasing need to think about post-trial analysis, even years post-trial, and the possibility of using these trial data for the purposes of exploration as an engine of future discovery. Ruthie is going to talk about study designs and how they're adapting to this sort of seeming conflict between research and discovery, and I'll step through these specific bullet points just to see how at least we've seen it playing out. So use of genomic data in trials is relatively new and not that frequent yet, all of your experience on the webinar aside. Just based on a quick clinicaltrials.gov search, about 500 trials a year have a genomics-related assay in the NAPCOM measure. About 100 of them seem to relate to genome-level sequencing. Don't take my numbers as being totally accurate. This is just based on some searching and some keywords that I came up with. But I think it gives you a general sense of we're not talking about tens of thousands here at this point, not yet. Most commonly, these are phase 1 and phase 2 trials, and based on quick numbers, about 50% appear to be oncology trials, with many of the other indications spread fairly evenly across the remaining 50%. And clearly, having single markers as inclusion-exclusion criteria is nothing new either. That's been done now for many years. Things like BRCA and breast cancer as a means of filtering patients is nothing new. For genomic level data, or even sets of genes in tandem for what's called a signature, these concepts are now appearing more in exploratory objectives or endpoints. I have listed here some examples of what we've seen in recent protocols, obviously de-identified and removed important terms so nobody would know what these, which protocols these are associated with. But breaking it down, you can look at it into different categories. In some cases, it's during the conduct of the study comparing survival curves such as Kaplan-Meier's against genomic markers, be it DNA or RNA. It could be evaluation of blood markers such as circulating tumor DNA as a surrogate for determining if there is cancer or as an early warning for tumor existence or metastases. It could be measuring DNA mutations in matched patients versus control tissue when available or separately as a marker of sensitivity to treatment, or measuring RNA expression for characterization of tumors or gene lists as a signature for efficacy. And these can occur either in the conduct of the trial or in the case of some of these larger studies you see from many pharmas like AstraZeneca and many others, where they will bank samples for later biomarker analysis using whatever assay emerges as time goes on. So what this leads to is a set of challenges when you get into the different phases I was talking about earlier. So we just talked about study design and new approaches that they have to use in the protocols. For an organization set up for sort of what you would call a standard clinical trial infrastructure from the last 30 years, this is a big change. So what impact do these assays have on trial execution? So on this chart, genomic data looks like one little bubble. Oh, you just bring it in. What's the problem, really? But in a way, it really does blur the line between research and development even more so as, in what was the fundamental question we talked about earlier. And the challenge is you either build a large infrastructure to run these assays internally with all the known IT challenges, or you outsource to new providers that you may not have worked with if you're new to this space. In terms of actually running what's in that bubble, there are multiple processing steps in the laboratory prep for the extraction of DNA or RNA, if this is something that your company doesn't do already, requiring specialists. You have to have the freezer infrastructure to hold the samples, which sounds mundane, but you have to be able to actually track the data and know what sample belongs to which subject and which visit it was taken from. And then, of course, you have to have knowledge of how to generate these data and run these assays. And once the raw data has been generated, which at the human genome level can be in the order of 100 gigabytes, exome level, maybe a tenth of that, but still not a small amount, you need a lot of storage space and you need a lot of computationally intensive ability to perform computationally intensive processes on CPUs that you have to deal with. And of course, there's much scientific debate about how to do variant calls and which reference genomes, et cetera, that you have to deal with. So each step can be a multi-hour process run serially if you don't have the computational resources that scale out, such as in the cloud. These siloed systems and these bioinformatics pipelines often don't link back to that clinical data that's already been captured in systems such as RAID. So it makes the linking of these data together even more challenging. And in addition to that, should be performed in a validated manner as well. Once the data is integrated, there's still a challenge in analyzing these data combined with the phenotypic data as well. Generally, there's different educational areas for bioinformatics, biostatistics, biometrics, and there's growing crosstalk in many organizations between these departments, but it's still relatively new, and it's a new communication thread that has to happen. Centralizing these analytics in at least large organizations is often not done, or it's being worked on, but it's difficult to try and get it all in one place. There's a lot of custom scripts, there's a lot of analysis inefficiency, and you don't really have things like audit trail built into software that you want to have, that you have in other parts of the process. Another difficulty is you have a lot of different stakeholders with different levels of knowledge. MDs, PhDs, experts in their domains, and then computational staff have to figure out ways to communicate these data at a summary level enough for the MDs and the clinicians, while at the MDs and the researchers, while at the same time conveying that you don't want to, let's say, from an observation, determine that it's a definite finding when there could be type 1 errors and all sorts of other problems that come up. Post-trial analysis, in addition, it has all of the challenges you would expect as well. You need to keep the data in a permanent place. It needs to be well enough annotated so that if someone else comes along and tries to analyze the data, they can actually go about doing that. But where there are challenges, there can be opportunities. And one scenario where this worked, and this is a cherry-picked scenario and totally biased, this is work done with a collaborator of mine. And just to highlight, though, it doesn't have to be genomics to be precision medicine. It doesn't have to be oncology. But it kind of leads to what kind of possibilities could be there if more data is collected from clinical trials, data sets, the SDTM data sets, the ADAM data sets, the genomics data that could be added on top. And this could be performed years after the fact. So just a brief use case on this. So there was a study done, rituximab in ANCA-associated vasculitis, called the RAVE trial, as a new approach in the induction of remission. It's a randomized, double-blind, double-dummy, active-controlled, non-inferiority study. And it was determined that rituximab was not inferior to cyclophosphamide treatment for the induction of remission in severe ANCA-associated vasculitis. Okay. And as it happens, and this data is in there is a clinical trials registry for this. You can read the identifier there. Putting out a plug to my old project. This data is also available at import.org at the row level. And this was work funded by NIAID, Genentech, and Biogen. So rituximab met the criteria for non-inferiority. Great. Now what's interesting is, in a paper that Mazen Nasrallah put out a few years ago now, he worked in Atul Butte's lab at the University of California, San Francisco. He downloaded the data from Import and thought, well, wouldn't it be interesting to see if I could further characterize these patient populations? So I can go into more of this in the technical Q&A portion if there's interest, or just download the paper. It's in Arthritis Research and Therapy, the citations at the bottom. But effectively, just taking flow cytometry data, which is commonly taken in clinical trials, he was able to, based on a new metric that he came up with called the granularity index, which is basically just a measure of granularity in the white blood cells, determine if for hyper or hypo granular cells, which treatment would be more effective. Now, granted, this is exploratory, so it has to be certainly followed up on, and further hypotheses have to be done. But this is one study, and they were able to find that the remission rate for cyclophosphamide and rituximab would both be better if you just used the metric of the GI to determine if you had hyper or hypo granulocytes. just based on side scatter effectively in the flow cytometry data. So again, assay run all the time. Imagine if you could expand this out across multiple studies and actually get better ends. Imagine if you had genomic data that could also characterize what's going on in that person's immune system at that particular moment, as opposed to just having flow cytometry data. Now, I apologize, I got one step off on my slide. But so this shows that decision tree I was just talking through. So as you can see, the profiled therapy in the middle is the granulocytes, hypo or hypergranulocytes going off into the decision trees into the right and the left, or rituximab and cyclophosphamide. I never say that, cyclophosphamide. So again, imagine if you could do meta-analysis. Imagine if you could increase those ends to more than 99 patients. Interestingly enough, since very few clinical trial data sets are available at the row subject level, there are few publications comparatively to the meta-analysis of trial data with the purpose of subsetting patient populations downstream. While in genomics, this is becoming increasingly widespread, although with less phenotypic information than they have in, say, a typical clinical trial. In resources like GEO or some of the EBI resources, it's not as well annotated in that respect because they're generally discovery studies, although there are exceptions to that as well. There was actually a great Outlook article in Nature last week regarding work in identifying blood biomarkers for mental health, infectious diseases, etc. In one case, for infectious diseases, which happens to again be what I've worked in a little bit more recently, they highlighted the work of Pravesh Khatri from Stanford, who has had success in identifying gene signatures for classifying viral versus bacterial infections, for also classifying sepsis and to the degree of severity of sepsis, tuberculosis in the inactive state, etc., all based on looking at publicly available genomic data. Pravesh does not have a single wet lab in his lab. It's purely a computational group. So imagine what you could do with cross-trial meta-analysis over time, especially if you start to get genomic data in. So Medidata, just to kind of step back from that whole clinical trial process we were talking about, has an extensive platform already for managing a clinical trial. The study design optimization process tools lead to more efficient and less costly study designs by removing redundant or unneeded tests or procedures. The unified implementation of RAVE with balance, RTSM, means fewer data points required for entry. Fewer patients may be needed with the use of what's being developed now as the synthetic control arms. Again, I could go on and on about all the different aspects of this. But the point is, Medidata is now expanding quite a bit into the data sciences and has hired up a very large team to try and figure out how to bring all these data together and how to analyze it from both an operational and, to some extent, a scientific point of view as well. So one of the modules we're building out right now is something called Medidata's Clinical Trial Genomics. As far as genomics and precision medicine, we're trying to build out an offering to help with some element of what we were just talking about. We understand that many of these different aspects of trial management and genomics can't be solved by one individual tool, but we still at the same time think there's some hand-holding and help that can be provided to reduce what has been quoted as 75% of the time spent in analyzing data being in the data wrangling, just trying to pull all the data together. So when it comes to CTG, the idea is that this is a product that's built as an element in the Medidata platform. We try and provide fairly user-friendly ways to ingest the genomic data that's already been generated, and that gets linked automatically to Medidata's clinical data sets, and then we can also support loading of analysis data sets. And for within there, we can then map and link the subject level data to the experimental results, perform some data checks that you couldn't perform before having the joined clinical and mechanistic assay results, genomic in particular, and then some dashboards for progress. So in theory, this would help during the conduct of the trial itself and try and bring the genomic data back into the everyday part of clinical operations, or at least into being able to know if these have been performed or not, and if any glaring issues have been found in the genomic data. And then on top of that, once the data is integrated, we can provide some summary statistics, some what I would call exploratory analyses, to help at least come up with initial hypotheses on perhaps ways you could stratify your patients or subset patients based on the joining of the clinical and the genomic data together, and ultimately other assay types as well. Just to step through them briefly, as I mentioned, this is part of the Medidata platform, so you get out of that scalable cloud storage and processing, you get role-based access, you get all the HIPAA security, all those sorts of things. In terms of the QC, we're talking about identification of quality problems such as sample and subject repeats. We're trying to determine in many cases if samples have been swapped. We could do things such as consistency between genomic data and covariate labels, such as subject sex. Also could get into things like tissue pattern matching to see if really what the tissue being specified matches a genomic signature, at least not for tumors, obviously, but in other cases. And then determining if there are outliers and any issues that might have been raised in a particular lab or a particular day's runs. And the idea is that these data can be fed back into the rest of the Medidata platform and then made available to the appropriate users at the appropriate time in the clinical trial process. In terms of analytics, again, we're talking about proven common approaches for patient stratification, clustering, integrating with survival analysis, regression, covariate detection to try and identify to filter down the fields of interest, regression trees. They can be run automatically or user-driven. We're trying to also make use of all the clinical data that we have to allow for filtering of patients and initiating these analysis, and of course running an audit trail so that in a multi-user environment, you can see who's been doing what sort of analysis. And just briefly describing the clustering method, and again, this is just a validation exercise, but one way we wanted to make sure our process was working was to validate against the triple-negative breast cancer patients in the TCGA data. So our process goes through and it does the data pre-processing step, log transforming expression data. This is all based on RNA-seq data. It does a dimensionality reduction using t-SNE. It performs the clustering using a standard method called HDB scan. Again, these are all published methods, nothing unusual here. And obviously outlier detection, and then identifies representative genes and puts them into different clusters. And interestingly enough, again, this was submitted to ASCO last year and was put out in the publication that accompanies it. But just really, this was a validation exercise for us to make sure that we could identify the triple-negative breast cancer patients without, obviously, those 3 proteins included in that. And interestingly enough, it was able to surface a novel genomic signature for triple-negative using all of the other genes. And finally, to get outputs similar to what I showed earlier from the RAVE paper, although the methodology was different, the visualization similar. Being able to use things like regression trees or recursive partitioning to try and bring together the clinical data and the genomic data, and to be able to at least get some initial hypotheses as to what clinical factors or genomic factors may be subsetting patients or might indicate something to pay attention to. We're very well aware that these can all lead to type 1 errors. We have every intention of making it clear to users who are not statisticians that everything has to be taken with a grain of salt. And a big part of what we're trying to do here is to ultimately enhance the communication between those different departments, the researcher side and some of the statistical and quantitative sciences side. But with that all being said, okay, so we've talked a little bit about those different boxes in the clinical trial workflow and how it all fits together, but it doesn't mean a darn thing if the FDA doesn't know what to do with it when they get it. And again, I haven't really talked about what trial designs are actually best suited to dealing with these targeted therapies. Whoa, sorry, I jumped ahead one too many. So the point is, now Ruthie is going to step in and talk a little bit about these areas. And so Ruthie, I'm passing it on to you.
Thank you, Jeff. And thank you to the audience for being here and giving us this chance to talk to you. I want to take just a minute and tell you a little about my background, since I will be talking a lot about the FDA today. I'm currently at Medidata in the data science group, but previously I worked at the FDA for more than 20 years. I started there as a reviewer, worked my way through the ranks, and at the time I left was a deputy division director in the Office of Biostatistics in Cedar. So I think, that gives me a certain perspective about what the FDA is thinking and doing. And so I'd like to share some of that with you today in the context of precision medicine. So just in general, I think it's a really exciting time to be working in the pharmaceutical development area. And we, as an industry, are presented with a great opportunity to utilize precision medicine approaches to transform, really, the way that products are developed. We will be set to answer multiple research questions simultaneously. No longer will a single trial answer a single question. These things will be posed and answered in unique trial designs, answering multiple questions at a time. And as recently as, this has been suggested by many people in the industry, but as recently as 2 weeks ago at the National Academies of Science, Engineering, and Medicine meeting, Janet Woodcock, who you probably know is the director of the Center for Drug Evaluation and Research at FDA, stated that the clinical trial system is broken. And Dr. Woodcock is a strong proponent of personalized medicine and alternate drug development strategies. And I think her leadership at the Office of New Drugs in Cedar will have a big impact on the implementation of these personalized medicine ideas. So we really are on the verge of having big changes in the way we do these trials, I think. But for now, let's start with talking about where we are today. The trials of today often enroll a highly varied population with the protocol inclusion and exclusion criteria designed to define the disease of interest, but also to allow one to cast a wide net for the recruitment of patients. There are large variations from one patient to the next in terms of all kinds of baseline characteristics, whether they be genomic or others. And we know that some patients will respond to the investigational product and some will not. And to some extent, we ignore this fact, calculate an average response for the overall group. This can lead to murky efficacy findings with the study level treatment effects being marginalized by the inclusion of the non-responders. In the ideal trials of tomorrow, we aim to be much smarter. Studies will start with the same highly varied patient group, but we will integrate clinical and molecular patient data to guide selection of the most appropriate target therapy for that individual patient. Patients will be classified into precise subpopulations for assignment to a therapy that's targeted to patients with those characteristics. And this will optimize the patient's efficacy and safety outcomes. So we know that patients often respond differently to the same medications, and this survey reinforces that fact. There are large segments of disease populations that do not respond to certain treatments. And why is that? One reason it could be because the patient's baseline characteristics put them in a category of patients who cannot receive benefit from a particular product. What this means is that patients may be at risk for possible safety events associated with the product and none of the efficacy benefits. And clearly, this is not a favorable risk-benefit profile. When we get smart enough to understand who these patients are and which drugs are targeted to specific patients and for whom they will work, we can avoid subjecting these patients to possible safety events, and all the while observing greater overall efficacy results in the particular subsets than would have been possible in the larger heterogeneous group. But how will we get there? I apologize. So I want to talk a little bit about announcement by the President in 2015 for precision medicine. The Precision Medicine Initiative was designed to revolutionize medicine and generate the scientific evidence needed to move the concept of precision medicine into everyday clinical practice. And of course, a natural consequence will be that it will change the way medical products are developed and evaluated by the industry and by the FDA. The 21st Century Cures Act allocated funds to NIH for precision medicine and biomedical research. Much of that is earmarked for research on brain disease and cancer. This legislation also modified the FDA review process, allowing companies to provide data summaries and real-world evidence in place of full clinical trial results in certain circumstances. This was somewhat controversial, but we can see FDA's response in several ways. For example, there's an ongoing initiative sponsored by the Clinical Trials Transformation Initiative and FDA to explore how real-world data can be used for registrational purposes. Although this recent legislation and announcement from the President has fueled attention to precision medicine, FDA has had a long-standing interest in precision medicine. The first FDA-approved targeted therapy was Herceptin for treatment of early-stage breast cancer that is HER2 positive, and that was in 1998. Since then, there has been much more activity in these areas with publication of several guidances related to precision medicine and the approval of many more targeted therapies. However, this is not without growing pains, and one of the significant stumbling blocks at the FDA has been that these products generally require multi-center policies and standards across CDER, CDRH, and CBER. In place now is the Companion Diagnostics Guidance, which describes development of companion diagnostics that are essential for the therapeutics product's safe and effective use, and HER2 positive triple negative PIK3CA tests to determine appropriate breast cancer treatment is an example. But things are not always as simple as a diagnostic test that is essential for safe and effective use. Often the safety or efficacy of a therapeutic is affected by a predictive factor, but not essential for its use. For example, better efficacy is expected in certain subgroups, but some efficacy is expected across the board. This concept is described in FDA's enrichment guidance, and strategies for using predictive markers to increase the efficiency of clinical trials for demonstrating efficacy are discussed. In addition, the adaptive design guidance lays out the agency's thinking on many study design and analysis methods that are helpful in investigating precision medicine. Adaptations such as modifications in the treatment randomization ratios, dropping or adding an arm, or revising a patient's treatment based on their inadequate response to an initial treatment are all examples of how adaptive designs can fuel precision medicine. Medidata's product Balance that is designed to randomly assign treatments and dispense appropriate drugs to appropriate site locations is well equipped to facilitate all these unique study design features. The co-development guidance provides advice for obtaining contemporaneous marketing authorization for a therapeutic product and its corresponding companion diagnostic. And expected soon is the complementary diagnostic guidance. This guidance is expected to lay out a framework that allows for a diagnostic device that aids in the benefit-risk assessment of the therapeutic product, but is not essential, as with the companion diagnostic. It is expected that development of a complementary diagnostic will be voluntary on the part of the sponsor, but will potentially allow description of the diagnostic test in therapeutic labeling. A recent article published in the New England Journal of Medicine and authored by Janet Woodcock and Lissa Lavange, who is the director of the Office of Biostatistics at CDER, discusses master protocols with applications to personalized medicine. Master protocols investigating multiple therapies, diseases, or both are described. The intent is to capitalize on similarities among trials, for example, shared control groups, recruiting efforts that are similar, etc. Woodcock and Lavange group these master protocols into three types: umbrella, basket, and platform. Umbrella trials study multiple targeted therapies for a single disease. Basket trials study a single targeted therapy in the context of multiple diseases or disease subtypes. And platform trials study multiple targeted therapies in the context of a single disease but in a perpetual manner with new therapies entering or exiting as the trial is underway. The lung MAP trial is a late-phase umbrella trial in advanced squamous non-small cell lung cancer. Patients undergo a common biomarker screening to classify patients into genetic subgroups where they are assigned an experimental therapy expected to be effective in that subgroup. Each targeted therapy is evaluated independently of the others. Some umbrella trials include randomized controls in the biomarker subgroups and some do not. Since the approval of nivolumab for advanced non-small cell lung cancer, the lung MAP protocol was redesigned as single-arm investigation. The article by Woodcock and Lavange suggests that the study of antibiotics for resistant pathogens may be facilitated by basket trials. Patients with varying types of infections are enrolled, screened for the target pathogen, and assigned to experimental therapy as a single-arm or randomized control investigation. The ADAPT protocol is one such example. I-SPY2 is an exploratory phase platform trial designed to investigate new treatments for biomarker-identified subtypes of early-stage breast cancer in the context of neoadjuvant therapy. It includes response-adaptive randomization to assign patients to the most promising treatment or combination of treatments in their respective genetic breast cancer subgroup, while maintaining a sufficient number of patients assigned to the standard of care. It has shared use of control patients and discontinuation of therapies or arms with low probability of success, and recommendations for advancing therapies or arms with high probability of success for further development. So with all of these guidances and publications, it's important to point out that FDA and precision medicine is not just talk. There is concrete results too. FDA continues to approve targeted therapies intended for certain subgroups or diseases. And this is just a sampling of some recent targeted therapies that have been approved. As you might expect, many of these are in oncology, but approvals do extend to other therapeutic areas as well. We've covered a lot of ground today. Jeff has described the discovery of biomarkers and how correlations among patients can lead to meaningful clusters for further study. We've talked about numerous indications of support for precision medicine from the FDA, including guidances, publications, and approval of targeted therapies. Precision medicine is positioned to revolutionize the way pharmaceutical products are developed. Multiple research objectives are beginning to be investigated in an efficient and targeted manner using master protocols. I believe the industry is on a path that will lead us to the goal of providing more effective and safer treatments through the targeting of specific patient subgroups with the most advantageous benefit-risk profile. So I think we have some time for questions now, and I'd like to turn this back to the moderator.
Thanks, Ruthie. We'll move on to the Q&A. Just a reminder to the audience, there's still time to submit your questions using the Q&A button at the bottom of your screen. We already have a lot of great questions, and we'll try to get to as many as possible. First up here, Ruthie, can you elaborate on what is the synthetic control arm and how that could be used?
The synthetic control arm is an exciting new product at Medidata. It's based on historic data, clinical trials data, that has been collected by the industry over more than 15 years. And this data has been collected through the Medidata electronic data capture platform. And Medidata has clinical data aggregation rights to use this historic data to facilitate further study. So the synthetic control arm is one use of that data. It takes historical patients from trials in a therapeutic area that is similar to a set of experimentally treated patients of interest, and we select the historic patients with baseline characteristics that match the baseline characteristics of the current day experimentally treated set. And in this way, we create a pseudo control group for comparison of those experimental results. It's intended for therapeutic areas where single-arm trials are the norm and a randomized control is somewhat unethical or impractical. And so it replaces a comparison, or replaces or enhances, I guess, a comparison to literature or a comparison to clinical intuition. Instead, it provides a group of clinical trial participants from similar previous studies, and it provides something that looks an awful lot like a randomized control.
Questions for Jeff. What is the validation status for the CTG product?
Oh, okay, sure. So we're in the process now of sort of completing version 1 of CTG through the next couple months, and we'll be going through the CFR 11 validation through next year. So that will probably about sometime into 2018, it'll be fully CFR 11 validated, and we'll go through and do a bigger launch of the product to the larger community.
Not sure if this is directed at either you, Jeff, or let's see, what parts of the Medidata platform can help execute adaptive trials?
I think I can take at least part of that question, and then Jeff, if you have additional comments. I think it's helpful to know that the Balance product is fully equipped to to adapt to the assigning the randomization and supplying the study medications for adaptive trials. So randomization ratios that vary, or studies with treatment arms that discontinue, or new treatment arms are introduced, or studies which allow patients to be removed from treatment arms when they're not achieving good outcomes and assigned to a different treatment arm. Those are all easily handled with the Balance product. And also, I should have probably mentioned with the SCA question before, the primary intent of SCA is to provide that control group. But another use that comes through with the creation of that control group is to investigate those historical patients and identify subgroups of disease where patients are doing differently. Either they're responding to treatments or a class of treatments in a different way, or they're having different adverse events. And in this way, generating hypotheses from the large historical database that's facilitated by the number of subjects that are there, we can generate hypotheses about subgroups of patients that really would have been impossible to see in a prospective study, just because the prospective study is often limited by, the possible sample size.
What other therapeutic areas have you seen utilize genomics in trials?
Sure, I guess I can take that one. So, go ahead, Ruthie, did you want to say something?
Well, I can comment, but I'd also like to have you comment, Jeff.
Sure, go ahead.
So as far as what I've seen, so in my experience actually at FDA, the vast majority of applications for personalized medicine are coming in through the oncology groups. And, they are, almost becoming commonplace, I think, in those settings, but they are really not limited to that setting. There have been a number come through in the pulmonary allergy group, and those were exciting applications. I had a small role in some of those, and it was a fun experience. Also, there have been suggestions for using these adaptive trials in the investigation of antibiotics. And even FDA was involved in a response to the Ebola crisis, and there were proposals for platform trials to investigate that epidemic. And so really, these are coming in a number of therapeutic areas.
Yeah, I don't have a whole lot to add to that either, but it's certainly in the immunology areas that I worked with in my last job, there were genomics assays being applied to certainly a lot of the observational studies and then some of the interventional ones as well, across asthma, across infectious disease, which we were talking about earlier, even, in some cases, across pretty much allergy, immunology, and transplantation, which were the areas that actually funded us on the last project. That was the division. But when I was doing the clinical trial search as well, pretty much the other 50% was spread fairly evenly across everything from cardiovascular to CNS, dermatology, GI, down the line, musculoskeletal, even nephrology. Now, these were hits for genomics in general, so it could have been lower throughput assays, so it might not have yet gotten into the next-gen sequencing kind of genome-level assays. But I think clearly it's getting more widely spread across.
Jeff, did you also ensure that you could replicate the RAVE trial data so you could trust the latter results you obtained?
Sure. So, and I should say, yes, I am technically an author on that paper, so that's a fair question. I'm in the back, not in the front, though. Mazen Nasrallah, who did the work, did a validation against the findings in the original publication, yes. And if you look that up, you'll see that in particular, he was able to reproduce the CD19 positive B-cell depletion graph that they had in the original publication. So the CD19 B-cell count versus days after randomization across the 4 groups in the study. And that is in both there's a chart in the original publication and there's a chart in the follow-up publication. And this was done as a way to validate the means of flow cytometry analysis that was done, which was sort of a new clustering method that didn't use two-dimensional gating.
Can you elaborate, Jeff, on the Nature paper you mentioned in terms of biomarkers and disease?
Sure. Yeah, I mean, I just was a reader like everybody else, but there was a paper, I don't have it right in front of me, but the author was Emily Sohn, S-O-H-N. The title was Diagnosis, Frontiers in Blood Testing in Nature Outlook. And I haven't, other than reading the review and finding it very interesting, that's about the extent of what I've done with it. But it does provide a nice summary of some different biomarkers being detected or different approaches for finding early detection of different disorders or classification of different disorders using just, in many cases, microarray gene expression data. And that's what, like I said, the Khatri lab spends all their time doing. And in terms of full disclosure, he's also a friend and a former collaborator. So again, I'm totally cherry picking based on my own experience. But it is interesting what they've been able to do in terms of classifying viral versus bacterial infections using these gene signatures. And he has a new gene test separating people with sepsis versus other inflammatory responses to a non-infectious cause. So, I mean, there's a whole litany of things in here, as well as other therapeutic areas where I'm not as knowledgeable, but they discuss liquid biopsies and probing the brain, et cetera, using blood tests. So, again, I could see how these sort of biomarker discovery areas could start to feed into clinical trials as well.
Ruthie, this question's for you. How harmonized are the regulations of adaptive design approach across different regulatory agencies across the world?
So I would say they're probably not harmonized. I don't even think they're harmonized within a single regulatory agency, at least speaking about the FDA. I think anytime you're embarking on an adaptive design, you need to have early communications with the FDA about how you plan to do that and gain their acceptance of those plans that you have. I don't think we've been doing adaptive designs or precision medicine long enough to say we have, a certain standard approach that should be expected to be okay in every setting. So I think, proceed with caution and talk to the agency early.
All right, this looks like this is going to be our final question. We had a lot of great questions today, couldn't get to them all, but we will be getting back to everyone who submitted personally after the webinar. So our final question is, can you elaborate, I'm sorry, I have the wrong question there. What is the difference between precision and personalized medicine?
That's a good one. You know, I think for better or for worse, they're used interchangeably for them in many cases. And I'm not going to claim to have a better answer than anyone else, to be perfectly honest. The way I think of it, and don't over-apply this logic, is that I look at precision medicine more as tailoring a treatment to an individual's or group of patients' characteristics. It doesn't mean necessarily you're creating a drug for an individual person, but it's more about classifying into subpopulations and kind of targeting therapy that way. While personalized medicine, I guess, anecdotally could mean coming up with a drug for that specific individual. So it's a bit of a fudgy answer, but you know, there's some subtlety and a difference there. I mean, I think honestly they're used fairly interchangeably, whether they should be or not. And frankly, what I probably should know is I should probably go back and find whomever came up with those two particular terms and see how they intended them to differ and be the same. Ruthie, I don't know if you have any other thoughts on that.
No, that's essentially, my thoughts are the same, is that many people do use them interchangeably, and I often think of them as interchangeable.
Jeff, Ruthie, thank you. I'd like to thank everyone for attending this Fierce Markets webinar and submitting so many great questions. I'd also like to thank Metadata for presenting today's webinar. A reminder that the webinar has been recorded. You will be able to access the recording within 24 hours using the same audience link that was sent to you earlier. Again, thank you for joining, and we look forward to seeing you at future events.